Hello. I'm the scientific founder of Deciduous, and you got a bit of a background from Sebastian through his fancy video, that talking about rejuvenation of the immune system, and we have a very different, not a different, but a particular perspective on rejuvenating the immune system to clear senescent cells that accumulate with age as well as in disease. So as I start, I want to give the disclaimer that I'm a scientist at heart, and I will talk a lot more science than I've heard in the past, couple hours. So, but it's fun science, and I hope you like it.
Anil, would it be possible to, yeah, exactly. You're headed there now.
Yes.
Perfect.
Okay, so Deciduous was formed about 3 years ago, and it came out of the work out of my lab, and this is the basis of what Deciduous thinks about and does. And the general idea is that healthy tissue has, due to aging or disease insult, the cells, a certain type of cells respond to the stress and become senescent. Senescent meaning not just the inability to proliferate and be young, but also they secrete a lot of harmful factors, cytokines, chemokines, and other things to essentially create havoc in their neighborhood. And usually they're there because the immune surveillance has failed to remove them. And if you do have proper immune surveillance to remove them, then you can go back to homeostasis and there is no disease. The problem really is with these senescent cells is when they accumulate is because the immune system has failed to remove them. And that's the key sort of basis of Deciduous Therapeutics, is when immune surveillance fails, there is accumulation of tissue. Now, when this accumulation occurs, it's the basis of a lot of diseases, and this has been active work in the last 5 to 7 years. And a number of diseases have been shown to lead to accumulation of these senescent cell types within different tissues. And this accumulation correlates with disease, and in many genetic models or in drugs that I will show you, removal of this leads to improvement in the disease outcomes. So the general concept really was that the immune system has failed. Let's try to mimic that and remove them and do the job what the immune system is failing to do. So that's turned on to the whole class of disease drugs called the Cenolytic drugs. These are drugs that were targeting certain pro-apoptotic pathways in these senescent cells, and essentially leading to their killing these cells and removing the cells and doing the job what the immune system fails to do. The problem with this approach is that a lot of the targets are commonly expressed in other cell types, and so there's a lot of side effects of using these drugs. So think about it as an analogy to cancer therapy. 10, 12 years ago, you had chemotherapy drugs that did target the tumor cells, but they had a lot of side effects. And think of senolytic drugs in that class. And then now think of immunotherapy, where you have exquisite targeting and very little side effects, where you target the tumor cells directly. So this is where Deciduous comes in, is we are an immunotherapy company for removing senescent cells. Since this is a meta-obesity and a focused meeting, I wanted to show you what the senescence does in a model of obesity and type 2 diabetes. This is just a quick transcriptional profiling at a single cell level from a comparison between mice that have been fed a high-fat diet, so their diet-induced obese mice, and regular age-match controlled chow mice. And you can see that there's many different kinds of transcriptional profile clusters, but you can see that the cells from the high-fat diet mice all cluster to one side of the, and that turns out to be, as you can see here, is that they all turn out to be senescent cells. So these are senescent cells that are seen only in the adipose tissue from the high-fat diet. This is probably the first demonstration of in vivo senescence accumulation seen so clearly. So what happens here is this is not truly a assay, but we had to develop an assay to really look at this, and so we developed a flow cytometry assay where we could essentially look at the presence of senescent cells in the adipose tissue when you give a mouse a high-fat diet, and you can quantify this and do it routinely. And once we did that, we could do a lot of screens to understand what is the immune regulation that's allowing these accumulation of senescent cells. So what part of the immune system is being inactivated so it prevents the clearance of these cells. And we focused on these molecules here, which is called the MHC class 1-like molecules. These are the molecules that are expressed on the cell surface that communicate to the immune system. And we identified, after a lot of screening, one particular kind called CD1D, which is an MHC class 1 molecule that is present on the immune cells, the antigen-presenting cells is what we call them. And this directly talks to one particular kind of in a T cell, which sits in between the innate and adaptive response, and it responds to a lipid antigen and gets activated. And this is the cell that has the ability to go find a senescent cell and kill it. So this part of this, what I don't show you, this part of this immune system with age as well as in the disease state is inactivated. And so our approach was then as to how can we then stimulate and sort of rejuvenate this immune system so that it does its job of finding the senescent cells and removing it as it's normally should be doing endogenously. The Deciduous has, made a lot of molecules that will do this specifically, and we did this so that we could actually formulate to deliver it as well in humans, as well as target the human TCR on the NKT cells. And so we have a whole series of molecules that do this, and here's an example of what it can do. So this is a diet-induced obese model, as I mentioned before, that we use to do as a preclinical study of our molecules. When we deliver this molecule, the 080 molecule from Deciduous, you can see that we now activate the iNKT cells, which are normally inactivated within the tissue. Unlike what Sebastian was talking about, the iNKT cells are not circulating cells. These live within the tissue. These are resident cells. And so the activation drug has to reach the resident tissues where they accumulate, where the iNKTs are activated, and that's what this drug does. You can see here. So you get target engagement by active expansion of the iNKT cells. And correspondingly, now when we use our assays to measure senescent cells, we can see a big drop in the senescent cells that were accumulating in the obese mice that are now removed. And with this, we get huge benefits, metabolic benefits. So insulin resistance, as measured by HOMA-IR, is now back to normal levels as to what it was. And this is within a week to 10 days after one single treatment of the iNKT activator. And also we have now the blood glucose, which is the blood glucose incursion you normally see. What's called a glucose tolerance test is now normalized to levels that you see in wild type chow-fed mice. Is this specific to metabolic disease? We have a whole different model for fibrosis in the lung, the IPF model, and very similar mechanism also. Now the same drug again activates the iNKT cells, delivery to a bleomycin-induced injury model. And you can see again a reduction of senescent cells in the lung, as well as an improvement in the fibrosis, reduction in the fibrosis with the treatment. And this is just done with one single treatment over the period of 3 weeks. So this is a very different way to treat disease, because we're actually going to the root cause of the disease, which is the accumulation of senescent cells due to the immune system essentially failing to do its job, and we are then re-stimulating the endogenous immune system that's and telling them to do their job that they're not doing. So unlike most drugs, this is a single treatment that is given periodically, is how we envision it, every couple months, and treatment for some things like type 2 diabetes or idiopathic lung fibrosis that doesn't currently have any proper treatment, has clearly what we would try to do in clinical trials. So generally, our approach has been, to summarize, to harness a particular immune cell and rejuvenate that immune population to do its job of specifically targeting senescent cells, and to remove these senescent cells leads to actual benefits in the disease outcomes. This is just a quick overview of what the press has been talking about this work. Our funding is on the left side here, and this is the team that, when they're not in the lab, it's a company that's fairly well built out until we go into a Series A for a clinical trial in the next coming 6 to 8 months.
Wonderful. Thank you so much, Anil. This is exciting. It's exciting data. What's the next major milestone besides funding for Deciduous?
Yeah, so the major milestones really are, we are about to declare a candidate, a DC, and we had to work out the formulation to really deliver it properly and to lead to a delivery that targets the resident iNKT cells, which we have solved, and to have a series of compounds that do work. They're also geared towards humans, and so once we do that, we will set the path or the clock towards the IND approval.
And do you have any imminent plans for a next funding round?
I think in the next 6 months or so is when we will start to raise for the, once we, go down the path of the IND, we will then, when there's a clear path, we will then go raise money for Series A.
Okay, wonderful. Well, thank you so much for the time and the presentation today. We'll be watching.
Thank you. So by the way, this is my email here if you need to contact me and get more science than you got today.
Thank you.